learning using state-of-the-art softwares and sophisticated technology. Thus, we are able to offer a service that equals, if not surpasses, face-to-face learning, and to lay the basis for a global virtual school. In addition, InnovaProfs offers an advanced learning management system that allows parents to monitor their daughter or son's progress and give and receive feedback. Well, during the session, all participants will be muted, and Mrs. Hoda will unmute you if needed. If any of the participants needs to pose a question or answer one, you can do that through the chat of this meeting. Now to introducing Mrs. Hoda Motawe. Mrs. Hoda has 22 years of experience teaching two parts, the theoretical and practical parts of biology. She has an MA in medical applied chemistry. She is a Cambridge examiner since 2009, and nowadays is a senior examiner. She has also some top achievers, ranking top world and top Egypt. She teaches Cambridge and teaches OL, AS, and A2. So now, Mrs. Hoda, the session will start. Thank you.
Good evening. Today I'm giving the free lesson introducing the IGCSE, the O-level biology. Okay. Before starting, I need to illustrate something about the syllabus code of the O-level. And also I need to share this with you, and I need to show you some, I'll talk to you about, first of all, the syllabus codes. IGCSE, or as we commonly say, O-level biology, now have two syllabus codes. The old one, which is 0610, I don't mean by old that it's not applied nowadays. No, it is still, and you can register for 0610. But here the grading system would be in the letters like A star, this is the top grade, A, B, C, and this is the passing grade. Okay, less than this, okay, so it means that you didn't pass it. Another syllabus code, which is 0970, and here the grading, the numerical system from 9 to 1, okay? In Egypt, we consider that 9 and 8 equivalent to A star. And if you get 7, this equals the A. 6 and 5, both of them equals B, but with different percentage according to Egyptian Tansik, okay? And the passing grade is 4, and this equals to C, okay? 4 is a passing grade. Less than 4, if you get 2, 3, 2, and 1, okay, this means that you didn't pass the O level. For the syllabus component, both have same components, same topics, same papers. You sit for 3 papers. Paper 2, okay, which is an MCQ paper. Paper 4, which is a structured questions paper. And there is a third assessment. You may have paper 6 or paper 5. In paper 5, you sit for a practical exam. In paper 6, you have something called alternative to practical. Those are the 3 papers you sit for both 0970, which depends on numerical system in grading, or 0610, which depends on the letter system in grading, okay? In some parts of the world, if you are joining an abroad university, 9 is the A star, 8 is the A. but in the Middle East, we consider 9 and 8 equivalent to A star, okay? In the O level, I'm going to talk about the contents of the O level, taken for example, the 970. You have something like 21 topics, as you see. First of all, you study the audio, you know about the characteristics of living organisms, okay? Or sometimes we call it the Mrs. Gren, okay? This is the start of our curriculum. And then we study together the classification of living organisms. We know about the different types of organisms, different kingdoms and so on. Then secondly, we'll study about organization of the organism, cells, tissues, organs, differences between them. Then we will go through some cell physiology, like we will know how molecules move in and out of the cell, biological molecules, namely carbohydrates, proteins, lipids, and newly we will know more about DNA as a biological molecule. Then we'll study about enzymes, okay, how they help the machinery and the metabolism within the cell. Then we will go through the plant nutrition. We'll study photosynthesis, okay, mechanism of it, what factors they depend on it. Then we will go through human nutrition. We'll study the balanced diet, contents of it, and how digestion go on through the human digestive system. And then we'll go through transport in plant. We'll know how materials are transported within plants, because here you have an organism. You will not depend on the simple movement of molecules. Also, we'll study transport in animals. After that, we will study the diseases and immunity, gas exchange system, respiration, okay, and then excretion in human, coordination response, drugs, reproduction, inheritance, variation, and selection, okay, the organisms in their environment, and a new science. It's not new, okay, for us nowadays, but this is the new way of studying medicine and treating diseases and diagnosis diseases. Depends mainly on biotechnology and genetic engineering, okay? In the end, we'll discuss the influence of human on their ecosystems. Let me start first with talking about characteristics of living organisms. I'll turn back to the note. Okay. When I say characteristics, this is the first part. Characteristics of living organisms, okay, they are 7 characteristics. And I will take the letter of each characteristic and give this abbreviation. Mrs. Gren. What is this? M for movement. Okay. R for response. S, I'm sorry, R for respiration, sorry. S for sensitivity. G stand for growth. R for reproduction. E for, this is number 6, excretion. N for nutrition. This is number 7, this is number 5. So here you have the 7 characteristics, okay? And again, we give an abbreviation of Mrs. Gren. We will talk about each and every one now, okay? For movement, if I need to define movement, the ability for organism to change its location or move, actually. Organisms need to move when they seek for food, shelter, or they respond to a stimulus. In respiration, and please try to discriminate between respiration and gas exchange. All organisms in respiration, all organisms respire, but not all of them can breathe. Please discriminate between the word respiration and breathing. In a matter of fact, a bacteria is a living organism, but bacteria doesn't breathe. It just exchanges gases to do the respiration. What is respiration? The process of producing, I will not say producing, of getting energy from nutrients. This process may happen in presence of oxygen, and in this case we call it aerobic respiration. Sorry. Undo. Undo, please. Undo. I'm sorry. We call this aerobic respiration. Sometimes this process happens in absence of oxygen, and in this case it's called anaerobic respiration. And we will come back to the difference between aerobic and anaerobic in the chapter concerning the respiration. Okay, insensitivity, this is the ability of organism to respond to a stimulus. Like, if a car is approaching to you in a high speed, what you gonna do? You're gonna run. You're gonna run away. You saw the stimulus coming to you, and then you move to run away from it. For example, in plants, when you use a plant pot and you put it beside the window, okay, you will find it after 1 or 2 days, most of the leaves, most of the plant have been oriented towards the window towards the stimulus, which is the light energy coming from the sun. Growth, I mean by growth, this is the irreversible, and I mean it, irreversible increase in size and mass of organism. Okay, in reproduction, this is increasing the process in which the kind increases. This happens sexually and asexually. In excretion, this is the removal of metabolic waste product. And we need to discriminate between excretion and egestion. Egestion is something different. Egestion, this is when you have undigested food, and this food have been processed by each and every part of your digestive system. And after that, it's not able to be broken down or absorbed by the cells. So what's going to happen? Your body gets rid of it. But this process called egestion. But in excretion, it is a different issue. I mean that the component of food have been digested by your body and absorbed, okay, and reached the bloodstream and have been used by the cell. And there are some metabolic wastes produced, so you need to remove it. You know what? When you exhale, when you breathe out, this is excretion. You know why? Because when you breathe out, you take away carbon dioxide away from your body. What is carbon dioxide? It's a product of respiration, and it's a toxic waste. And you need to remove it, because if it is kept in your body, it will harm it. Okay, for nutrition, this is the obtaining of food. And we will come up to nutrition, we will know that there is an autotrophic nutrition and heterotrophic nutrition, okay? Now, we went through the Mrs. Grant thing. Now you know that there are 7 characteristics. Without those characteristics, I cannot classify the organism as living organism. For example, you know, nowadays we are isolated in our homes because of a virus, the corona, the COVID-19, okay? You know, virus is not a living organism. Virus is just something made of DNA and protein code. Why I did not classify it as living organism, okay? Is there any question now, Susan? Hello, Susan, are you there? Okay, is there any question? From Adham, okay, you said that they are, they cannot reproduce. Yes, Adham, okay. Right, again, this is a good answer, Adham, okay. I return again. Why viruses are not classified as living organisms? I have to bear in my mind the Mrs. Glenn. Viruses, as Adham said, he told me that they cannot reproduce Adham, they can reproduce, but don't feed. They don't excrete. They don't grow. They don't respond to a stimulus. Okay? They just can reproduce and they just can move. Abu Bakr, they do not grow, but they can mutate, yes. They can mutate inside their host, ya Abu Bakr. Okay? They can mutate inside the host. Out of their host, they cannot reproduce. Okay? And actually, what happens that under certain circumstances in the cell, which gives them the ability to mutate, okay? They don't decide to mutate. Okay, Abu Bakr? Okay. Again, let me start with the building block of any living organism, which is the cell. Of course, not all of us can see a cell by their naked eye. They just can see it using a tool called light microscope. Okay, we went through this. Okay, we will see that cell structure using electron microscope and light microscope. In a light microscope, you can only see what's in front of you. What do you see? Can anybody answer me? What do you see now? Huh? I need somebody to pick my question. What do you see? Nobody's there? Screen share is not working? Okay, I'm escaping. End the show. Zoom.
Excuse me لو في اي مشكله في السكرين شيرنج عند البارتيسيبانتس عند الطلبه بعد اذنكم اعملوا ليف للسيشن وريجوين اجين. شكرا. Miss Hoda, you can go on again.
Okay, thank you. Is there any problem now? Mr. Muhammad, do you see my screen?
No,
You don't?
no, the screen is not working now.
Yeah. Again. Share. You see my screen now?
Yes, the screen is clear now.
For everyone?
If there's a problem with the screen sharing for anybody, please type. Okay, it seems it's clear for everybody now.
It's clear now? Okay. I returned back again. In this sample, you have a sample of onion epidermis. Okay. And it has been stained so that the components of the onion epidermal cells are clear. And we use a light microscope. What do you see now in this image? What could you see? Can anybody answer me? Nobody? Okay. All you can see now is having a cell wall, as you see now, here, Here is the cell wall. And you see a nucleus and nothing else. Do you see anything else? Again, there is another cell here. This is the cell wall. And just the nucleus. And what's around the nucleus? What is so-called cytoplasm. Okay? If you are using a sample of animal cells like cheek cells, again, here plant cells, sorry, animal cells don't have a cell wall, they have a cell membrane. And here is it. This is a sample of cheek cells. Here it is. This is one cheek cell, have a cell membrane, and we have a nucleus here, and what we see here is the cytoplasm. Okay. But if we use electron microscope, we will see more details. Electron microscope gives you more details. You will see that the animal cell we were observing now, you see, I'll return back to the previous slide. See here, I just see a cytoplasm, I just see the nucleus, I just see the cell surface membrane. But if I use an electron microscope, I see more and more details. What do I see? I see nucleus, very large, as you see now, here it is, much better, larger than what we saw in the other diagram. We will see that surrounding a nucleus, this is what's so-called the nuclear envelope. This is the nuclear envelope. Also, you see some components like network of membrane. This is a network of membranes called endoplasmic reticulum. Sometimes you see it as rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, why sometimes we call it rough? Why sometimes we call it smooth? Because you see these black spots here on the endoplasmic reticulum. You see all these spots? Okay, those black spots are our so-called ribosome. These are the workers which help us to make proteins in the cells. Okay, also you can find some organelles, okay, like the mitochondrion. Mitochondrion, this is the site of aerobic respiration. Okay, here and there you can see another organelle called the Golgi Body, okay, and some organelles called lysosomes. In the chapter of diseases and immunity, we will study more about lysosomes and their function. Let me go to the next slide, because in a plant cell, you see another detail. You see here at this part, this large part here, okay, of course, this is, here is your nucleus, the largest organelle in the cell. And there is a large part here also called this plant cell vacuole. In the vacuole, you see, it contains the components of stored materials. For example, you see the flower petals, sometimes they are colored red, sometimes they are colored yellow, sometimes they are colored pink, and so on. Where are these pigments stored? Inside the vacuole. Vacuole also the place where some mineral ions are stored inside it. Another organelle, which we didn't see in animal cell, we usually see it in some plant cells, okay? which is the chloroplasts. And we will return to chloroplasts individually. We'll talk about it, and we'll talk about its function and where you can find it, okay, in the next coming slides. Okay, this is a generalized plant cell. Again, you have details, you see it when you use electron microscope, like the nucleus, like the mitochondrion, ribosome, the vacuole, as I said, Golgi apparatus. Sometimes you'll find chloroplast, smooth endoplasmic reticulum, and rough endoplasmic reticulum. I will go for one very important organelle, which is the mitochondrion. Mitochondrion, this is for single, but in plural, we call it mitochondria. Okay, mitochondrion has two membranes, outer smooth one and an inner folded one. Okay, as I told you before, mitochondrion, this is the site of aerobic respiration. You know why we have folded one inside? Because on this membrane, we have a very important enzyme called ATP synthase. Oh, what is this ATP thing? ATP is what's so called, ATP is the energy currency of the cell. Any metabolic reaction inside the cell is fueled by ATP. So mitochondrion is the place where ATP is made. On those inner membrane, you will have an enzyme called ATP synthase. Enzymes are the machinery that catalyze the formation of this ATP. The more folds we have, the more those enzymes we have, the more ATP are produced. Okay? So function of mitochondria is aerobic respiration. Mitochondria appear using electron microscope, okay, in this way, okay? This is when we use electron microscope, we will have this image. I return again to a sample I have taken from some plant leaf cells, and I used light microscope to help me observe the components of a plant cells inside the plant leaf. All I found here okay, again, this is the cell wall of the plant cell, and we notice that there are a lot of green circles, and I don't see any details within it, okay? Those green circles, each one is called chloroplasts. I mentioned that this is a sample taken from plant leaf cells. I can stop now to take some questions. Is there any question here? Anybody have a question? Okay. So again, I mentioned that this is a sample taken from a plant leaf. From your previous study in the core syllabus, okay, plant leaf, this is the factory where nutrition happens. This is the place where photosynthesis syntheses occur, okay, precisely inside this organelle, which is the chloroplasts. Let me go to the next slide to talk more about the chloroplast and how do they look like when I use electron microscope. Oh, usually, by the way, in electron microscopes, every image is appears like black and white. You see here, I can see more details inside the chloroplast. I will return to the previous slide. Using light microscope, do you see any details more than having green patches and green circles? Okay, nothing. But when I use electron microscope, I will see that there are a lot of details inside this chloroplast. Okay, like you see those lines or these dark areas, these bands. This band is made of membranes. Each membrane is called thylakoid. On those thylakoids, you will find pigments called chlorophyll. Oh yeah, chlorophyll, this is the pigment that can absorb light energy. Surrounding the chloroplasts, these thylakoids are stack of those thylakoids are called granum. Okay, many thylakoids together makes a structure called granum. Surrounding these grana, we have fluid called stroma. Okay, because photosynthesis needs part where light energy is absorbed and another part that can make contains some intermediates and some molecules that help formation of glucose. Okay, later on we will study photosynthesis in detail in the chapter of plant nutrition, and we will return to those, okay, in the chapter concerning the plant nutrition. Again, I returned to the cytoplasm where I can see rough endoplasmic reticulum. Rough endoplasmic reticulum is network of membranes. Those membranes, okay, stuck to it a structure called ribosome. So what is the function of rough endoplasmic reticulum? In the cell, there is a protein factory, okay, which makes, assembles the building unit of a protein and makes the whole protein. This is called the rough endoplasmic reticulum. So function of rough endoplasmic reticulum is synthesis of proteins. These molecules need a lot of steps, okay, so that we end up by formation of such proteins, okay? Every cell produces a huge and different types of proteins, okay? This is because some proteins work as structural proteins and some are functional proteins. Like enzymes that help us in digestions, structurals like collagen that makes your skin, keratin that makes your hair. All these are protein needs to be made inside the cell. Which part of the cell responsible for protein synthesis is the rough endoplasmic reticulum. Ribosomes, we will go through its structure. We discovered that it's made of small unit and large unit. Together, all these together are called ribosomes. And this is what we found on the surface of rough endoplasmic reticulum. This is how rough endoplasmic reticulum looks under electron microscope. Another organelle, which is important, very important, okay, is called Golgi Apparatus. Why should I have Golgi Apparatus? Sometimes proteins are made, okay, again, this organelle is called Golgi Apparatus, or sometimes, or you can call it Golgi Body. Okay, why a cell needs the Golgi apparatus? You see, again, it's a system of membranes, okay, I'm going through it, and have near to it some vesicles. In some cells, we need to secrete proteins. Golgi apparatus help us to modify this protein and pack it in vesicles and secrete it outside the cell. You know, all of us in our mouth contain saliva. Saliva contains a molecule called mucus. Okay, this mucus is made inside your salivary gland cells and then secreted to the outside. Okay, who help us to modify the mucus protein and secrete it outside the cell? The Golgi body. So function of Golgi body is to modify packaging of proteins and secrete it outside the cell. Okay. Is there any questions so far? Hope so. No questions? Another function of Golgi apparatus is making of an important organelle, which is the lysosomes. Lysosomes are organelles that contain hydrolytic enzymes. You know what? Sometimes they call lysosomes as suicide bags. Lysosomes help us to get rid of worn out material inside cell. Lysosomes have a function that help phagocytes after engulfing bacteria okay, to break down the bacteria completely. You need, some sort of immunity that when the phagocytes swallow the bacteria, okay, as you see here up there, okay, in this image, you see that lysosome fuses with this bacteria, and those enzymes started to break it down completely and get rid of it. This is one sort of immunity. We will go to this, okay, at the chapter of diseases and immunity, okay? These are the components of a cell so far, but you need to know something which is very important, that not all cells have same shape and functions. Sometimes cells need to differentiate okay, according to function. Till this moment, we have the information that each cell must have a nucleus, a cytoplasm, some organelles like rough endoplasmic reticulum and smooth endoplasmic reticulum. You must have a mitochondria. You must have Golgi body and lysosomes. What else a cell can have? Okay, there are some modification happens according to function. And we will study some examples, like ciliated cells in trachea, phagocytes. This is a sort of white blood cells in your blood. Red blood cells, a sperm. And we'll study some examples in plants, like the root hair cell, Xylem Vessels, okay? So let's see together how a ciliated cell differs from a typical animal cell, and how a phagocyte differs from a typical animal cell, as well as the red blood cell, as well as the sperm, and again, how a root hair cell differs from a typical plant cell, which is the palisade cells. Palisade cells also are differentiated cells. But before going through such things, I will stop for some time, okay? So that I can get an answer. So, sorry, I get some questions. Anybody has a question? I'm waiting. Ahmed, you have a question? Rahma? Or Jana? Or Menna? Or huh? So it seems like everything is clear so far. Rahma, everything is clear so far, ya Rahma?
Yes, Miss. Everything is clear.
You don't have any problems so far? Okay. طيب, should I go on? Should I go on? Okay, some specialized cells. I will start with some examples of animal cells, like ciliated cells in trachea. You know what's the trachea? This is the windpipe. It is located in your gas exchange system. Okay, the gas exchange system has the windpipe, which is the trachea, and this is the bronchi, and it goes through your lungs. Trachea, we call it air passage. And air passages, okay, have its important function of clearing air from dust and bacteria. Okay, so when I take a sample of ciliated cells, I found out that they are shaped like this. Okay, an animal cell we were studying now, it just contained the nucleus, a cell membrane, and a cytoplasm. This is what we call a typical animal cell. But here, in a ciliated cells and trachea, now the shape is different. Now I have another component, which is cilia. Cilia are some type of protein fibers. Why they have those cilia? So the cilia is there to help wafting mucus that contain dust and bacteria. Every time the air goes in here, it contains dust and bacteria. Okay, what helps us to get rid of those? The beating movement of the cilia. Where are those cilia? Are in ciliated cells. Okay, right. Another example of differentiated cells, which is the red blood cell. A red blood cell which is found in your bloodstream has an important function. Which is transport of oxygen from lung to tissues. When I take a look on a red blood cell, I'm gonna be shocked. This cell has no nucleus, only have inside it an important red pigment, which is hemoglobin. And when I take a look on this cell from side view, a side view of red blood cell shows that it is shaped like this, biconcave disc. Why this is so? It is a biconcave disc, okay, to increase surface area for absorption of oxygen. Okay, so now what happened in the red blood cell? Has no nucleus and it has a biconcave shape, okay, disc. This is to increase the surface area for absorption of oxygen, okay? Now, we see that not all the animal cells must have this typical thing, okay? Sometimes they have something extra like a cilia, and sometimes they may lose the nucleus to fill this all the cytoplasm of the red blood cell with hemoglobin. This is an important, very important molecule, okay? This is a huge protein which fills all the cytoplasm of the red blood cell, okay? Because hemoglobin can bind to oxygen to make a molecule called oxyhemoglobin. Okay, type. To increase its surface area, it is shaped like a biconcave disc. This is to increase surface area for the absorption of oxygen. From where? From lung, and delivered to tissue. Without oxygen, our tissues, our tissue cells cannot live. Okay, right. Another differentiated cells in our body, and it's very important cell, which is the type of a white blood cells called phagocyte. When I look of a phagocyte under light microscope, I will see it irregular like this. Not only this, the nucleus is lobed. Okay, all this is nucleus. It is large and have many lobes inside it. Okay, not only this, if I examine it with electron microscope, I will find that phagocytes contains a huge number of organelles called lysosomes. Why? Because lysosomes, organelles, contain hydrolytic enzymes, okay, that helps the phagocyte after they engulf or take in the bacteria inside it, they break it down completely, okay? Another differentiated cells like the sperm cell. This is one of the sex cells produced by men. A sperm cell has a head, a neck, and a tail because sperm cells move. Okay, now look, those cells have a different shape from those of phagocytes, have a different shape from those of ciliated cells. Now we see that differentiation helps to do their function. Sperms must have their tails to swim towards the egg so that they can fertilize it. And we will return again to the detailed structure of sperms and eggs in the chapter of reproduction. Okay, not only animal cells have differentiated cells, plant cells also, okay, have differentiated cells, like a palisade cell. A palisade cells, they are in plant leaves. Inside the palisade cell, of course, a plant leaf cell has a cell wall and the cell membrane. And the nucleus and the large central vacuole. This is inside each and every plant cell, but they have also chloroplasts. Why? Because function of palisade cells is to make photosynthesis. To do this, they have a large number of chloroplasts. But let me go down in the, in the plant. You know, plants typically have roots, stem, and leaves. Let me go here and examine inside the root. Actually, in the outer part, okay, in the root epidermis, the part that faces the soil, you know, around this root, there are a lot of soil particles. And take a sample of one cell from here. I'm going to be shocked. Why? Because this is the shape of what's a so-called root hair cell. Can anybody tell me what is the function of root? Huh? Who can answer this question? What is the function of a root? Can I pick one at random? Adham. Huh? What is the function of a root, Adham?
They absorb water and minerals from the soil.
Okay, what else?
Anchor the plant in the soil.
Anchor the plant to the soil. Yes, Adham. Thank you, Adham. Okay, so to do this function efficiently, okay, root hair cell extend here a finger-like projection. Okay, you know why? This is to increase surface area for absorption. If I examine the thickness of cell wall, I will find it very, very, very thin cell wall. So why thin cell wall? This is thin cell wall. This is to increase, actually decrease diffusion distance. Okay, so as Adham told us now, root hair cells are for absorption of water, absorption of mineral iron, and not only this, also anchor the plant to the soil. Okay, so to do this, it has this finger-like projection. Okay, this what help anchor the plant to the soil and increase the surface area for absorption. Okay, thin cell wall. I'm sorry, the pencil is not clear with me. Okay, but it has a thin cell wall. Okay, this is to decrease the diffusion distance so that mineral ions and water can diffuse faster into the root hair cell. Okay, another differentiated cells are the xylem vessels. When we examine a xylem vessel, our xylem cells, we'll find out that these are dead cells. No nucleus, no organelles. And before differentiation, xylem vessels were located above each other in this way, but after differentiation, cell wall between them are disappearing, okay? Not only this, wall of xylem is lignified, okay? Means what? That the cell wall contains lignin that prevent collapse of xylem vessels. Right. Why there are no organelles, no nucleus? So that you have a continuous and less resistance, continuous and no resistance for movement of water. Xylem vessels are the vessels that transport water and minerals from where? From root up to plant parts. Okay, so of course, this is why we don't have end walls here, and also there are no organelles, no nucleus. Okay, this is to give a continuous movement of water column and no resistance for the water column inside xylem vessels. Is everything clear so far? Everything is clear so far? Nobody speaking my question? Yes, everything is clear. Jana is clear. Absorbs nutrient. Okay, good girl, Jana. Okay, thank you, thank you, thank you. Okay, I will stop the lesson, or you want me to explain more? I go ahead. No, yes, carbon dioxide is carried in the red blood cells, but mechanism of carrying carbon dioxide inside the red blood cells, you're gonna study it in the AS biology, inshallah. Okay, I'll scroll the questions. If anybody has a question, please type it now. Rahma asked if carbon dioxide is carried in the red blood cells. Yes, carbon dioxide is, some of it is carried in the red blood cells, and there is some sort of reactions happen inside the red blood cells to carry the carbon dioxide. But details of this you are going to study in the AS biology. Any questions? Adham, you have everything clear? Yes, miss, okay. Who else may have a question for me? Is there anything you need to ask about the details of the IGCSE? Okay, I hope you enjoyed the lesson today. Okay, I'm willing, okay, to see you in a future lesson, okay, so that we can complete the curriculum together. I hope it's useful for each and every one of you. Okay, Thank you very much for joining me today. I hope to see you once again. Bye.